Good morning. We're going to talk about Chapter 17 now, where we're looking at gene expression. In other words, what gene manufactures what protein. And of course, we all know that genes code for information. They contain specific nucleotide sequences. This is the DNA we're talking about. And as you know, DNA codes for specific traits and directs protein synthesis. And we know that proteins are going to be involved as well as RNA molecules. Proteins are between the genotype and the phenotype. Now, gene expression is the process by which DNA directs the synthesis of proteins or RNAs, because we got to still make our messenger, ribosomal, and transfer RNAs. And that protein synthesis is going to involve transcription and translation, as we talked about in our protein synthesis video. In 1902, Garrett discovered that genes dictate the phenotypes of organisms through enzymes. And he theorized that the symptoms of an inherited disease reflect the inability to either make a particular enzyme or make an effective form of that enzyme. For example, alkaptonuria causes black urine because of alkapton, and it darkens when exposed to air. And there's no enzyme in these people for its breakdown. Later, this was called the one gene, one enzyme hypothesis, where a gene dictates the production of a specific enzyme. One gene, one enzyme. And so we know this was very important because this led us to a better understanding of metabolic pathways where specific enzymes controlled all steps, each and every step. In the 1930s, Beadle and Ufrusi theorized that Drosophila, that's fruit fly, remember, eye pigment synthesis was blocked by a lack of an enzyme at a specific step. Beadle and Tatum worked with bread mold Neurospora, and they won a Nobel Prize for this, by the way. Worked with x-rays and nutrition of this and proposed the one gene, one enzyme theory. So they worked with wild-type Neurospora. They grew it on minimal media that contained inorganic salts, glucose, vitamins like biotin, and agar. And when it was grown on minimal media, the wild type could produce the other things that it needed. And the colonies were all made of identical cells. Now, they discovered some nutritional mutants, and each of them was unable to synthesize a particular essential nutrient, and they can't grow on minimal media, which doesn't contain those nutrients. Remember, the wild type could produce what it needed, and these nutritional mutants cannot. The nutritional can grow on a complete medium where you add all 20 amino acids and a few other nutrients. And they said the gene for the enzyme to make a nutrient must be disabled in order for this to happen. Serb and Horowitz used mutants to study arginine synthesis and came up with 3 classes of mutants, and each blocks a particular step to catalyze the enzyme. Then this theory became modified because not all proteins are enzymes, obviously. For example, keratin. They're structural materials that make up hair, or insulin as an example of a hormone. So it was revised to read one gene, one protein. And what's interesting is many proteins, though, are made of 2 or more polypeptide chains. And so you have to have your own individual gene for each of those chains. For example, hemoglobin is made from 4. So then it was restated, 1 gene, 1 polypeptide, which is still not entirely accurate because a eukaryotic gene can code for a set of closely related polypeptides, and this is going to depend on how it is spliced. For example, in our protein synthesis video, we were talking about our pre-messenger RNA and how you could remove the introns and splice together the exons. Well, what gets removed determines what polypeptide you're forming, okay? And also, there are a few genes that code for RNA, which is also important in cells, but this never gets translated into protein. These gene products called proteins versus the more accurate polypeptide. So keep in mind that now we're saying 1 gene, 1 polypeptide. Now, please watch the video on protein synthesis for more specific drawings of what we're getting ready to talk about. It occurs in all organisms, includes transcription, which occurs in the nucleus. It's where we're making messenger RNA from DNA, and the DNA serves as the template or the pattern, followed by translation, which occurs in the cytoplasm on a ribosome and involves the synthesis of a polypeptide following the directions provided by messenger RNA, and this occurs at a ribosome. Now, if we look at bacteria versus eukaryotes, bacteria obviously have no nucleus, so they have no nuclear membrane. And what's interesting about that is they can begin translation with no processing as soon as transcription is finished. In eukaryotes, they do have nuclei, and this separates transcription and translation not only in space, transcription in the nucleus, translation in the cytoplasm, but also in time, because we have to wait for the pre-messenger RNA to be modified and matured before it will be exported through a nuclear pore. And remember, transcription in the nucleus, messenger RNA is going to the cytoplasm for translation. The messenger RNA is modified to become fully functional before it leaves the nucleus. So the pre-mRNA is processed into finished or mature messenger RNA. So the primary transcript is what we call the first messenger RNA that you get from the gene, including those that make RNA and not translated into proteins. Now, Crick in 1956 stated the central dogma, where DNA is going to make RNA, is going to make protein. And this framework is still generally accepted by scientists. In the 1970s, there was an exception. We discovered some enzymes that use RNA as their template for DNA synthesis. So this creates what we call a triplet code. So these codes in messenger RNA are called codons. They're made out of 3 bases, 3 nucleotides, and they will equal 1 amino acid. And there are 64 possible non-overlapping codes. In transcription, 1 strand of DNA is used as a template, and this is determined by how the enzyme is oriented. It transcribes genes, and this depends on the particular DNA sequences that are associated with that gene. So remember, messenger RNA is complementary to DNA. So as far as the nucleotides go, you have ribose as the sugar, uracil instead of thymine. Of course, all the other bases, adenine, guanine, and cytosine, remain the same. And the new RNA is synthesized in an anti-parallel fashion. The triplets are called codons, and for messenger RNA, they're going to be written in a 5' to 3' direction. These are also used for DNA nucleotide triplets along the non-template strand, identical to messenger RNA, except they're, instead of using a U, the other side of the DNA will use a T for thymine. And this is called the coding strand, not the code. Translation: the codons are decoded into an amino acid sequence, and the codons are read from a 5' to 3' direction. Each codon is made out of 3 nucleotides and codes for 1 amino acid. Now, excuse me, in the early 1960s, we finally cracked the genetic code. And this is the one that you had in the lab handout. The first was done in 1961, Nuremberg, UUU for phenylalanine. All 64 were done by the mid-60s. 61 of the 64 we discovered code for an amino acid. 3 provide a stop signal that helps the cell know when to stop transcription, and they are UAA, UAG, and UGA. AUG excuse me, has a dual function. It codes for the amino acid methionine. It's also the start codon. And MET may or may not be removed later when processing occurs. There is redundancy in code. Now, what does that mean? It means that if you look at that code, you'll see that for many amino acids, the only thing that changes is the 3rd nucleotide base, which means that it will code for multiple amino acids. It'll code for the same amino acid using multiple codes that differ only in the 3rd base. There is no ambiguity, though. Excuse me. You must be able to read in the correct groupings, and that's a grouping of 3 that make up the codon. And this reading section is called a reading frame, and it is read 5' to 3', and it does not overlap. This genetic code that we're talking about is pretty much universal for every organism. Genes transplanted from one species to another can actually be transcribed and translated. And this is wonderful evidence for evolution. For example, one of the best, most exciting things that's happened is biotechnology. And an example of its use is we can now use bacteria to generate human insulin by inserting the human genes for its manufacture into their genome. And that's really exciting because before that, we were using cow and pig insulin, and even though they were mammals, some people still had negative reactions to them. Now, let's talk about transcription specifically. RNA polymerase is an enzyme that forces the 2 strands of DNA apart and joins complementary RNA nucleotides to the template strand. And this is going to allow the assembly of a polynucleotide 5' to 3'. Remember, it always has to add to the 3' end. Now, this can start from scratch. In this particular case, as you're building an RNA, you do not have to have a primer. You do have the use of a promoter. And the promoter is a DNA sequence where the RNA polymerase can attach and initiate transcription. You also may see a terminator, but this is in bacteria only. That terminator signals the end of transcription in bacteria only. And remember, the direction of transcription is always called downstream. The other direction is called upstream. For example, the promoter of DNA will be upstream from the terminator. That's the way that you would use those terms. A transcription unit includes DNA downstream from the promoter is transcribed into RNA. Now, RNA polymerase is, of course, very important. In bacteria, there's only one RNA polymerase that synthesizes all the types of RNA. In eukaryotes like ourselves, there are 3 or more RNA polymerases. RNA polymerase II makes our pre-messenger RNA. All the other RNA polymerases transcribed RNA molecules that do not get translated into proteins. The stages of transcription include initiation, elongation, and termination. And we'll talk about those in part 2.